Millions of organisms inhabit planet Earth, many of which may never be discovered. Despite the vast diversity of life, all these organisms, both large and small, share one essential piece to their very existence: the cell. Much has been learned about the cell, but nothing compares in importance or magnitude to our knowledge of the cell cycle. The cell cycle regulates the growth, specialization, and replication of a cell. In this presentation, we will focus on the replication of the cell, which is exhibited in the process of mitosis, and, from a DNA standpoint, in the process of meiosis. Mitosis is the division of the nucleus and of the cytoplasm, resulting in two identical cells containing all the genetic information of the original parent cell. The ability of organisms to reproduce their cells through mitosis is the major characteristic that best distinguishes living things from non-living matter. For this presentation, we decided to use our favorite candies to better illustrate the phases of mitosis. Before mitosis even begins, the cell goes through interphase. During interphase, the cell's nucleus contains a nucleolus and duplicated chromatin that are tangled up so that you're unable to see the chromosomes individually. Cell division starts with mitosis. There is one diploid cell. Once prophase begins, the chromatin becomes more tightly coiled, thus separating the chromosomes and making them visible. Now there are chromosomes consisting of two sister chromatids attached by centromeres. The nucleolus disappears and the meiotic spindle begins to form. During prometaphase, the nuclear envelope starts to break apart, and now each of the two chromatids of each chromosome has a kinetochord. The kinetochord microtubules are attached to each centrosome. Next is metaphase, which is the longest stage of mitosis. The centrosomes are at opposite poles, and the chromosomes light up on the metaphase plate, which is an imaginary plane that is in the center of the two poles. Each chromosome's centromere lies on the metaphase plate, and each kinetochord of the sister chromatids are attached to the kinetochord microtubules, coming from the opposite poles. During anaphase, which is the shortest stage of mitosis, the two sister chromatids from each pair suddenly break apart. Now each chromatid is considered a chromosome. Each chromosome moves towards the poles along the kinetochord microtubules, which are attached to each centromere. Telophase occurs when the two daughter nuclei form in the cell. The nucleolus reappears, and the chromosomes become less dense. By the end of telophase, the cytoplasm is beginning to divide. In animal cells, cytokinesis involves the cleavage furrow forming, which pinches the cell in two. This marks the end of mitosis. You now have two diploid cells that are identical to the parent cell. Meiosis is the process where the cell passes on its nuclear material to its daughter cells. It maintains a species chromosome count during the sexual life cycle and contributes to the genetic variation of the species. Meiosis I separates homologous chromosomes, while meiosis II separates the sister chromatids. The first step in meiosis I is prophase. During prophase, the chromosomes begin to condense. Each sister chromatid pairs up with another forming homologous pair. Crossing over occurs when there is an exchange of DNA molecules on the segment of non-sister chromatids. This is called chiasmata. At the end of prophase I, the microtubules of each pole attach to the kinetochords, and then the homologous pairs move towards the metaphase plate. Next is metaphase I. The pairs of homologous chromosomes line up on the metaphase plate with one chromosome in each pair facing a pole. Anaphase I occurs when each pair of homologous chromosomes separates. The homologs move towards the opposite poles along the spindle apparatus. Even though the homologous chromosomes are now separate, the sister chromatids remain attached. At the beginning of telophase I, each side of the cell has a complete haploid set of replicated chromosomes. Cytokinesis takes place when the cytoplasm divides and forms two haploid cells using a cleavage furrow for animals. At the end of meiosis I, two haploid cells form, and each chromosome still consists of two sister chromatids. Meiosis II begins with prophase II. During this stage, the spindle apparatus once again forms, and the chromosomes move towards the metaphase plate. At this point, each chromosome is still composed of two chromatids attached to the centromere. In metaphase II, the chromosomes are lined up on the metaphase plate. The two sister chromatids of each chromosome are not identical because of crossing over. During anaphase II, the sister chromatids separate. The chromatids, which are now considered chromosomes, move towards each pole. In telophase II, the nuclei form, and the chromosomes become less dense. Cytokinesis once again allows a cleavage furrow to form, and separate the cells into four haploid daughter cells, each with an unreplicated set of chromosomes. Each of the four daughter cells is genetically different from the other daughter cells and its parent cell. At the end of meiosis, you end up with four cells that contain half the chromosomes of the parent cell, 23. This is a major difference from the two meiotic daughter cells, which each have 46 chromosomes.